All right, so we have seen a sequence of different experiments over 200 years that attempted to wrestle with the idea of spontaneous generation of things like maggots and then things like bacteria. And what these experiments ultimately needed was a way of controlling both the physical presence of bacteria and the hypothetical presence of vital forces and corrupting gases. And if it hadn't been for the ability to do that, we would still believe in spontaneous generation today. Okay, so the next thing we're looking at is epidemiology. And we teach this in microbiology a lot, to show how we understand infectious disease, and these are some amazing stories. Epidemiology is the study of disease patterns. It can be anything from injuries, like in epidemiology journals, I've read articles about roller skating injuries and risk factors in roller skating. But also epidemiology would be how do epidemics spread, or what controls outbreaks of diseases. Epidemiology is an old field. One of the first examples we have is from Ignaz Semmelweis. He was an obstetrician in Vienna, and he famously, well, you'll see, he had a flash of inspiration and saved many lives in an early example of epidemiology. So he was a high-ranking obstetrician at a teaching hospital, and there were two clinics that would deliver babies in this hospital. One had medical students and one had midwifery students. In both clinics, 5% of mothers died from essentially an infection they got during childbirth. So this was around the same time that Pasteur was doing his experiments in the 1850s and 1860s. This was before the germ theory of disease was widely accepted. Indeed, while Pasteur was doing his amazing experiment with the swan-necked flask, most people in the world were unaware of it. As late as 1910, many educated people still believed in miasma. But scientists and people who were reading scientific literature would become more and more aware of Pasteur's work. So by the 1900s, well-educated people were aware that spontaneous generation had been disproven. So Semmelweis and his colleagues in Vienna were in the middle of this. This was happening around the same time, and they were completely unaware of what Pasteur's experiments would lead to. And they definitely didn't know that microorganisms caused this disease. So let's get into what Semmelweis saw. He collected data. What he noticed was that mothers who gave birth outside the hospital didn't die of that infection. They didn't die of puerperal fever. And so he wanted to see what was going on in his clinics that was risky for mothers contracting this disease. He asked people in his clinics, or ordered them to do things in a similar way so that he could compare them. And what he saw was that in the medical student clinic, almost 10% of mothers died of this infection, where in the midwifery clinic, it was more like 3%. In other words, a woman's risk of death from this infection was 3 times higher if medical students were delivering the babies. So what was the difference? Were the medical students incompetent? Well, presumably no, because mothers giving birth outside the hospital would have been dealing with less competent people, but still not getting that infection. The difference was that medical students conducted autopsies on mothers who died of puerperal fever, and then they went and did vaginal examinations and delivered babies. And since they didn't know really what bacteria were, and they didn't know that bacteria caused this disease, and they believed in spontaneous generation, they did not have any sense of asepsis or antisepsis. They didn't do things like wash their hands because they didn't know why they would need to. So this is kind of horrifying in hindsight. The midwifery students, on the other hand, they didn't do examinations of corpses. They didn't do autopsies. They also didn't wash their hands. They didn't have any information that the medical students didn't have. They didn't know anything the medical students didn't know. But the medical students were doing something horrifying without realizing how horrifying it was. So Semmelweis noticed this, and he was trying to understand how could it be that a corpse could somehow cause a disease using a medical student as an intermediary. So somehow the corpse being examined by the medical student would infect the mother examined by the medical student. But what's the connection? And he didn't know what the connection was. He had a breakthrough when a medical professor was cut by an autopsy knife and died of something like puerperal fever. What that made clear was that whatever it was in the corpse that was infecting people didn't need a human to carry it. It was something intrinsic to the corpse. And so what Semmelweis wanted to do was prevent anything from the corpse reaching the mothers. So he asked his students to wash their hands with chlorinated water after they did autopsies. And the students never would have done this if they hadn't been ordered to. This was a silly idea to them. And to other doctors, this would have been a completely silly idea. But once they started doing this, their rate of losing mothers to puerperal fever went down to equal the rate in the midwifery clinic, more or less. And over the 10 years, something like, I've read something like 3,000 fewer deaths happened at that hospital than would have happened otherwise. So he was directly responsible for preventing that many fatal infections simply by ordering people to wash their hands. Notice that it took him years to publish his observations, and unfortunately, two different things worked against him. One, he was obnoxious and would never tolerate criticism. No one liked him, so he had no charisma. But also, he couldn't explain what was going on. He could say what he did, he could say what his intervention was, but he couldn't say why it worked. So he couldn't convince anyone that what he did made sense. So no one read his publication, and obstetrics didn't change. He left the hospital to go to a lunatic asylum. Presumably he had some kind of condition that was destroying his brain. And after he left, the medical students stopped washing their hands, and puerperal fever returned, went up 6-fold. Right. So that was Semmelweis doing what we think of as the first example of epidemiology, where he looked at a disease pattern and found a variable that seemed to control it, controlled that variable to prevent infections. So that was that was Semmelweis. I guess the first modern epidemiology, the first one that was, I guess, well known at its time, was John Snow's ending of a cholera outbreak. So cholera is a disease that happens when people don't have access to clean drinking water. So typically if fecal material can contaminate drinking water, cholera is a likely disease that will start to occur. And there was a bad outbreak in London in 1854, where in the Soho region, that neighborhood was growing in population faster than sewage management could handle it. So people would have to use cesspits and latrines and things instead of sanitary sewers like what we have now. And this outbreak ultimately killed 600 people. London residents mostly believed it would have been caused by miasma. Again, 1854, this is before Pasteur's swan-necked flask, and this is London, not France. Pasteur was French. So these people believed that it was caused by a miasma. Well, John Snow is famous for interviewing residents and asking them about their daily habits. Where did they live? Did they get cholera? Didn't they get cholera? And he made this map where people who, well, he'd draw these little bars, one little bar for everyone who got cholera and lived at this address. And so he was able to notice that the center of this outbreak was right around here. So he was able to think, well, that must mean something. And he looked for what would it mean? What could be around here that would spread an outbreak of cholera? And he realized that there was a water pump that all these people were using as their source of water. And he used that map that I just showed you to convince local authorities to prevent people from using that water. And what they did was they removed the handle from the pump. Famously, I believe you can still find that pump in London because it is now famous. And that ended that outbreak. Snow eventually figured out that the epidemic started with a cholera-infected baby. Or this was his understanding. This baby's diapers were washed into a cesspit right next to the drinking water well. And so now we would understand that bacteria from the baby's feces would move through the groundwater into the drinking water, and then people would ingest them, the bacteria would grow in their guts and cause cholera. That's disgusting. You probably noticed that. And that's exactly what anyone he talked to would have thought. So local authorities just would not believe that a diaper could cause cholera. Instead, they thought that miasmatic theory was less disturbing, and they believed it. And it directly contradicts their evidence, but evidence doesn't change most people's minds. Humans, you're probably aware of this, but it's well known in psychology that our brains make decisions and then we look for reasons to think we are correct. We are not rational creatures. We're not logical creatures. It's very difficult to become a scientist and to force yourself to look at data all the time instead of believing things and finding rationalizations for them. That's what your brain wants to do. And this is a clear example of that. Okay. That psychology stuff, I don't explain it well, but it is fascinating. And scientists are just like anyone else. We have exactly the same kind of mental processes everyone does, but part of our discipline professionally is looking at evidence and letting evidence push us towards conclusions the evidence leads to. And that's just not how our brains are programmed to work. Okay, so 2 more people to look at. One of them is Joseph Lister, and he's famous. There's a mouthwash named after him. And he was a surgeon who realized he was losing a lot of his patients to post-operative infections. And he was aware of a lot of the research that we're talking about, and he would have known about what Pasteur had discovered. And he believed that there was a connection between microbes and disease. So what he tried was spraying carbolic acid on operative wounds. And what he found was that he was able to save his patients and prevent a lot of postoperative infections, prevent deadly infections by spraying this horrible chemical on their wounds. Phenol, this chemical that he sprayed on their wounds, if you're wearing latex gloves, it can go through the gloves and give you a chemical burn. Phenol is terrible. But yes, it would kill bacteria. Lister could both show that his intervention had an effect on the health of his patients, and he could explain what was going on using Pasteur's work. So he was able to convince other people to do this same type of antisepsis, and we still do today. And years later, obstetricians began doing the same thing. They started using antisepsis and preventing puerperal fever. They didn't do it because of Semmelweis. They did it because of Lister. None of these people had ever heard of Semmelweis. He just couldn't become famous because he couldn't convince anyone of anything. So, cautionary tale, be nice to people and be charismatic if you want to change the world. At least if you want to be someone in epidemiology. Let's see. Okay, and the last person we're going to talk about is Robert Koch, and he is the person who nailed down the germ theory of disease. He developed an experiment or a set of experiments that clearly implicate a microorganism in causing a disease. What you have to understand before you look at these experiments is the concept of pure culture. This is a thing in microbiology where if you have a tube with pure culture in it, every cell inside that tube is of the same type. So they're all the same species, or they're all the same strain. They're all descended from one cell that you put in there. That's pure culture. And that's really rare in nature. In nature, we have mixed cultures, where if you see bacteria, it's a mixture of different types of bacteria. So Koch developed what we call Koch's postulates. These are experimental results. If you do several experiments in a sequence, particular experiments, you can convince the scientific community that a certain microorganism is the main cause of a certain disease. So if that's what you're trying to do, you do the experiments Koch put together and invented. And we use this all the time in microbiology research. And what we say is that we have satisfied Koch's postulates. We have done these experiments, and we have shown that the microorganism is both necessary to cause the disease and sufficient to cause the disease. It is the thing that causes the disease. And that's that's entirely consistent with the germ theory of disease that says that infectious diseases are caused by microorganisms. Anytime someone satisfies Koch's postulates, they are strengthening the germ theory of disease. We can't do this for every infectious disease for a lot of different reasons, and we'll get to those reasons later. So here are Koch's postulates. The same microorganism is always present in every case of the disease. So he'd be thinking about a disease like anthrax that kills sheep. And anytime you find sheep that have died of anthrax, you always find the same microorganism. You always find Bacillus anthracis in those sheep. You have to be able to isolate those bacteria in pure culture. So you have to take bacteria from a sick sheep that has anthrax and get them in pure culture. Then microorganisms from that pure culture, you put them into a healthy sheep that doesn't have anthrax, and it gets anthrax. Then you take microorganisms from that newly infected animal and compare them to the ones that you got from the other infected animal, and if they're the same, you have satisfied Koch's postulates. Koch was studying anthrax, and that is really lucky. So he was brilliant in doing these experiments, but he was lucky because he had no way to get pure culture bacteria, except that in a sheep, Bacillus anthracis grows in the blood in pure culture. So he could take a pure culture of these cells and grow them in the laboratory, and they would stay in pure culture. So that's what he did. And if you go back a few slides here, if you read these little things, this is showing his sequence of experiments where you'd find a sheep with anthrax and you'd get the bacteria from it in pure culture. And you grow them up, inject them into another animal, and it would get anthrax. You then take bacteria from that animal, grow them in pure culture. They're the same bacteria. Well, you have satisfied Koch's postulates. These bacteria cause that disease. Right. So Koch was brilliant, but he was also lucky because anthrax is probably the single easiest disease to do this with. Pasteur was trying to do the same thing with cholera, but he couldn't because he couldn't get Vibrio cholerae bacteria into pure culture. So there's that. Okay, well that's it for history of microbiology. That took a long time. It's a lot of videos. In the past, I found that students have kind of struggled with these experiments, so I've taken my time explaining them, and hopefully they've made sense to you. And a lot of this is kind of fundamental to what we do in the lab and what we think about. So I'm glad if you've taken the time to understand. And from here, we'll go to microscopy and then a few other topics. And that will be fun. I'll see you there.